Hi everyone, welcome to the lab. As you can see, I am all set up and ready to go. Today we're going to be working on chromatography. This is a method that is used to physically separate any sort of chemicals to identify the parts of a mixture, and it's really done by size using gravity to separate. Food dyes are often a mixture of pigments that reflect different wavelengths of light. That means as white light comes down, many of the wavelengths of other colors go into the actual pigment, while the color that we see actually bounces off. So red pigment would absorb all colors except for red. People are very particular about the colors of the foods that they eat, so we often add pigment. We don't necessarily know which pigments are added unless we do a process called chromatography to separate those chemicals. In chromatography, we force these chemicals to go through a filter, which in this case is going to be paper, against the pull of gravity. That means that the smaller pigments are going to be able to move faster and the larger pigments are going to be very small. This is how we're going to separate each of the pigments by chromatography. So here you can see all of the materials for a simple chromatography experiment. First, we have the solutions that we're going to be separating. They're Kool-Aid and they've been mixed with water so that they will be easy to separate. We have our chromatography solvent, which is a mixture of ethanol and salt and water. And we have our simple chromatography equipment. We've got 2 Erlenmeyer flasks and 2 test tubes and 2 stoppers. We also have the chromatography paper, which is going to be what we use to actually filter the dyes so that we can separate them and identify what they are. In order to get the pigments to move away from one another, we have to have a part of our experiment that's going to be moving. This is going to be the solvent, or what is known as the mobile phase. We're going to draw the chromatography equipment and label where the solvent can be found. The only part of this equipment that I actually need to draw is the test tube, because the Erlenmeyer flask is there just to hold the test tube in place. The mobile phase is the solvent, which I am drawing down here at the bottom of the test tube. This is where the solvent will sit. It's actually going to be soaking up into the paper. It's going to move up the paper, which is why we call it the mobile phase. Once the solvent is in place, we are going to take a piece of porous paper, meaning it's loosely woven and has lots of tiny crevices where the solvent can enter and climb up. And on that paper is going to be the pigment mixture. So as the solvent goes up the paper, it is going to begin to carry with it the pigment mixture. And if it is heavy pigment, it will stay close to the bottom because it's going to be difficult to move, and the smaller ones will move up the paper further. So the first thing that I did was cut my chromatography paper. I actually have several pieces of chromatography paper here. I cut them so that they would fit into the tube, excluding this round portion at the bottom because this is not going to fit down there, and allowing enough room at the top so that my stopper would fit in. If my stopper is not nice and snug, my chromatography solvent is going to evaporate. So now what I need to do is I need to set it up so that I can put my solutions in place. So I am going to create a starter line about 1 centimeter, actually usually about 2 centimeters up from the bottom. So here I am with my ruler, and this is about 2 centimeters. Now you'll notice that I'm using a pencil. The reason is because the graphite in the pencil will be unaffected by the chromatography solvent. If I use pen actually gets dissolved by the solvent and is going to create a problem for me by adding additional pigments outside of the pigments in my solvent. So here I have my 2 pieces of chromatography paper, which have been marked, and my 2 solutions. And what I'm going to do is I am going to take a paper, it's basically a paper stick that will soak up the solution, and I am going to very carefully put it along the line so that the dye stays under my starter line. That's going to be where the starting point is for my dye. And I'm going to let it sit there and dry for a moment. And I am going to do the same thing over here on my second piece of paper, my second piece of chromatography paper. Now I'm going to let it dry for a moment because right now the dye is kind of holding itself in between the fibers of the paper, and I want to make sure that I have as much dye on there as I possibly can. The more pigment that I have on the paper, the more likely it is that I'm going to be able to distinguish between the dyes most accurately. If I don't have very much, it's not going to show up after it moves. So I'm going to go ahead and I'm going to take the dye again, and I'm going to carefully put it underneath the line. And you can see it does go a little bit above the line, but not by very much. That's a small amount of scientific error that we're going to have. So here's my next one. Now I'm going to do this until I have 2 or 3 applications so that I know that the amount of dye in there is sufficient. Okay, so I've finished putting my solutions onto my chromatography paper and given them adequate time to dry. I did a pretty good job putting them on there because I think that the dye is dark enough that we'll get some good results. So now I need to set up my 2 chambers. My chamber is actually going to be the test tubes. The vapor is going to stay in there, as well as the liquid form of the solvent. And we're going to use the Erlenmeyer flask to just hold them up. That's really all the Erlenmeyer flasks are going to do. Now to create the chamber, since the test tubes have openings, we've got 2 stoppers in place, and that's going to keep all of those vapors inside. Now for right now, I'm going to open them up, and I'm going to add 5 milliliters of solvent. This is a graduated pipette, which means that every time I squeeze up here, it is taking in 2.5 milliliters. And I'm going to put 2 dropperfuls in each one of these. Okay, and you can see them down at the bottom, right? So this is what it looks like. You can see the solvent down there. And right now it's starting to evaporate. But what I'm going to do now is I'm going to place the chromatography strips into each of the chambers, and then I'm going to put the stopper on top. Not so tight that I can't get it out, but tight enough that it's going to hold the vapors in. Now I'm going to bring this over so that you can take a look at what this looks like. Oh, here's the camera over here. So if you look at this, you can see, or may be able to see, that the solvent is going up the paper. Now that is important because as it hits the dye, it's going to begin carrying the dye upward. Now this is going to sit here for a little while, generally about 20 minutes, although it's kind of cool in this room, so that means that the molecules are going to move more slowly. So it may take closer to about a half an hour. And when we get done with that half an hour, we'll see what our results look like. These are the directions that go through what you just saw in those previous few videos. Okay, it's been about a half an hour, and I know that the solvent line has gone all the way up to the top, so the solvent front is very close to the top of the paper. Now, because this is ethanol in there, I want to protect my eyes, and I want to protect my skin. If you think about this as being about 30% stronger than hand sanitizer, it's not hugely damaging, but I certainly don't want it on my skin. So I'm going to use a forceps to do this. I'm going to take the stopper off. I'm going to remove the paper, and what I'm going to need to do is, because the solvent line is going to dry right away, I'm going to use a pencil and mark the top. And then once I've done that, I'm going to put my stopper back on, and I will return the chromatography solvent to the container in just a few minutes. I'm going to take out the second and lay it down, and again, I'm going to mark the solvent front with my pencil. And then I'm going to let it dry for just a couple of minutes so that I don't get any ethanol on my hands, and then I'm going to do the analysis. Once the paper is in place in the chromatography equipment, the solvent will begin to move. The edge of the solvent is referred to as the solvent front, and it will constantly be moving against gravity towards the top of the paper. As it passes the dye, it's going to pick the particles of dye up and carry it through the paper like a filter, with the heavier pieces making it up to the top and the smaller pieces staying down towards the bottom. You will be calculating the RF value, which is the retention factor of the paper. It's how tightly does it hold that dye in place. In order to calculate this, we are going to need to calculate the distance each food molecule moved and the distance that the solvent moved. We're going to use our initial line that we drew with the pencil as our starting point always, even though there is some paper down below. In order to measure the solvent distance, we're going to measure the distance between the first pencil line and the solvent front. For all of our RF values, this is going to be the number that is down at the bottom. Because there are 4 different pigments in this dye, we will have to calculate 4 separate RF factors, and you can see the distances have been indicated here for each of those lines. To calculate the RF for the red pigment, I will take its distance in centimeters and divide it by the distance that the solvent moved. For RF for pigment B, I will take the distance that that pigment moved and divide it by the solvent distance. We will repeat these calculations for the last 2 pigments. When you get your RF factor, it should be a number that is less than 1, so a 0 point something. If you get something that is larger than that, it means you're doing your calculations incorrectly. However, you might want to think about what an RF factor of 1 actually means. So now we're ready. Let's take a look at our results. First, you're going to circle the large area that each of the colored dyes seem to be in. Now you'll notice in the middle of the pink one, there's this big, what looks like a purple splotch, and that's because that is blue dye that for whatever reason kind of wasn't allowed to move ahead. Once you have each of those circled, then right in the center of that circle, you're going to draw a line. That line is going to represent about the average of how far the dye has moved. So first you need to measure the solvent distance. You need to line your ruler up with the initial line where the dye started and measure the distance in centimeters to the solvent front, which is the line that you drew after you took it out of the tube. To find the RF value for the red dye, you're going to need to measure the distance that the red dye moved. So you're going to line your ruler up with the initial line where the dye started, and you are going to measure the distance to that center line that we drew in the middle of the circle of red dye. Again, you will be measuring this distance in centimeters. To calculate the RF for the blue dye, you're going to have to measure the distance that it moved by lining up your ruler with the initial spot where the dye was and measuring the distance in centimeters to the center line of the blue dye. Now you're going to calculate your RF value. You can see the example here for the RF for the red dye. The distance that the red dye moved, 4.3 centimeters, is divided by the distance the solvent moved, which is 7.8 centimeters. And when this is divided, we get 0.55. That is our RF value for the red dye. You can use this calculation as an example as you go ahead and calculate the RF values from the lab. Now that you have your RF values, you should be able to go ahead and interpret these and find out the name of the food dye that was actually used. You're going to need a combination of 2 pieces of information, the color of the food dye and the RF value of the food dye. These things need to be used in combination to make your decision. Because chromatography is affected by environmental factors, there is a large range in which this food dye can actually fall into, and in some cases it may fall slightly outside of that range, so you may have to make a few judgment calls. But go ahead, look at your results and try it out for yourself.